Start recording. Hi, everyone, and welcome once again to the HICCC Resources for Research Seminar Series. Thanks for joining. Today, we're going to hear from Kenneth Olive and the group who run the Oncology, Precision Therapeutics, and Imaging Core, otherwise known as OPTIC. So we're very pleased to have them here. Just a quick introduction on Ken. He is, as I said, the director of the OPTIC shared resource. Ken got his bachelor's in biology from Bucknell University and his PhD from the lab of Tyler Jacks at MIT. He then went on to do his postdoctoral training with David Tuvison, both in the University of Pennsylvania and the University of Cambridge. Ken is an associate professor of medicine in the Department of Medicine in the Digestive and Liver Diseases Division. He is a NCI NIH funded researcher and his own research focus is on understanding the biology of how pancreatic tumors respond to therapeutic intervention using mouse models. If I got any of that wrong, I apologize. We're delighted to hear from Ken today. I will hand over to him. Thanks very much, Ken.
All right, thanks very much, Emer. Happy to share the activities of our core facility to the rest of the crew here. And I am going to give a number of examples today of kind of research applications, most of which have come from my own laboratory, just because I wanted to, not be randomly publicly sharing other users' data without asking them. But obviously the applications go well beyond pancreatic cancer. And I also want to tip the hat to Chris DeMosi, who I know is on the line. I don't know if the other members of our core are here, but Chris is the manager of the facility and the one that is, kind of the public face and engages the most with the users. And Chris is always happy to take questions and inquiries and just answer ideas. And the one thing I want to reinforce more than anything else is that, imaging and the other activities that we do, sometimes not every biologist is aware entirely of what can be accomplished. And so rather than coming to us and saying, I'd really like to image with this technology, could you teach me how to do it? We really encourage our users to come to us and say, these are the questions that I have biologically, scientifically, that I'm trying to answer. How can imaging help me answer those questions? And then we'll have a conversation, we'll get into it. All right, so the Oncology, Precision Therapeutics, and Imaging Core, OPTIC, which we're very proud of our acronym, has three main areas of service, and these are broadly broken down into the generation of personalized models, the conducting translational therapeutics studies, so actually running drug studies on mouse models for users, and then small access and utilization of small animal imaging technologies. And these can be mixed and matched. So sometimes a translational study will have an imaging component or will make use of a personalized model. The idea is that they're all supposed to be self-reinforcing. I'm going to give some vignettes today about the personalized models approach and about the small animal imaging. But just to very quickly touch on translational therapeutics in this one slide, the idea here is that there are many laboratories whose main focus is not running and utilizing the most sophisticated animal models out there or any animal models whatsoever. And so nevertheless, we want to make it possible for users to perform gold standard translational therapeutic studies. And so what we envision is basically having almost an a la carte menu where a user can come to us and say, I'd like to treat a model of this disease using this drug at this dose at this schedule, get these samples, look at this imaging, and then at the end, we provide back a publication quality data set that they can go ahead and use even if they've never touched a mouse in their entire lives. And so as examples of types of studies that we can run, we have pharmacology assays, maximum tolerated dose, pharmacodynamic studies, so the difference between pharmacokinetics and pharmacodynamics. Pharmacokinetics is what the body does to the drug. Pharmacodynamics is what the drug does to the body, and we can help you assess both of those things by setting up specialized study structure designs to get the materials and the samples you would need to assay and answer those questions. And of course, efficacy studies as well. So let's go ahead and jump into generation of personalized models. Over the past year and a half, 2 years or so, Chris has worked very hard to get this, to set up a pipeline that enables users to come to us and say, look, I really would like to generate a patient-derived model either from this specific individual patient that has interesting features, or patients that meet this criteria, say a patient that has a colorectal adenocarcinoma, or a uveal melanoma, or a patient that was a smoker, or et cetera. So we can go ahead and work with the database shared resource to identify patients that match the criteria that users are interested in, and then work with and then they will also help us with the patient consent process. OPTIC holds both an IRB and an HICCC protocol for this process, so you don't even need to get your own IRB protocol to do this. And then working with the molecular pathology shared resource, we can acquire the tissue from the patient as they undergo surgery. And right now we're limited to just doing surgical samples, but we're actually in the process of amending our IRB to expand that to include biopsy samples as well. And then going from there, once they hand off the tissue to our team, we can help make a variety of different types of models. And that could be generating a cell line, it could be generating organoids, it could be generating a patient-derived xenograft. We are also in the process of building a models bank. And so we do where we would do some characterization of the models, collate all of the clinical data in a de-identified way, coded so that our core facility can link back to the original data set if necessary. But then we actually strip the identifying information off of all of the annotation and samples that are associated with that model, and then can provide those to users. So we're basically running interference so that you don't have to get an IRB in order to go ahead and have a model to work with. And ultimately, our goal is to have a PDX bank that covers dozens of different cancer types, such that eventually someone can come in and just say, I'd like to go ahead and start up a mouse model, a patient-derived model of X, and we'll already have X in the freezer, know a lot about it, and can then even tell them, all right, well, this tumor has mutations in genes A, B, and C, and we can then implant it and run studies very rapidly. So from a single patient-derived tumor, we have the potential to generate a variety of different types of samples, including viable tumor fragments, primary cancer cell lines, snap frozen tissue fragments, organoids, which actually I forgot to put on here, biobanking, and then we bank those and potentially have the ability to do molecular profiling, and then ultimately the goal would be to get to the stage where we can do personalized drug screens. And I'm going to show you an example of that from my own laboratory. So we're beginning to build a pancreatic cancer PDX bank specifically with my laboratory, and we have about 10 models now that have been established. I'm pretty sure that we have a near 100% engraftment rate at this point. Pancreatic cancer is known historically for having a high engraftment rate, but our core facility does take a particularly aggressive approach in implanting tumors into orthotopic sites. It's not possible for every tissue, and sometimes you don't know where what the actual site of origin is of a given tumor. But when possible, we work very hard to try to implant in the native environment of that tumor, and we find that gives a slightly higher engraftment rate for a lot of cancers. And so the idea here is that one could create a bank of, get explants and create a bank of xenograft models, and then actually do various types of precision medicine analyses. In this case, OncoTreat from Andrea Califano's group, predict drugs and then test them in the expanded versions of those models. As an example, this is a single pancreatic tumor that came from a patient with pancreatic ductal adenocarcinoma. On the bottom left here is histopathology from the original primary tumor of that patient, and then 3 successive first passage PDX models that were derived from that tumor. And there are a fair number of similarities. There are some distinctions as well, of course, because we're going into an animal recipient that is compromised for its immune system. So the composition of the stroma is going to be different compared to the primary tumor. And you can see that even, just histopathologically, that the stroma is looser and more hypercellular than the original tumor. So there are going to be similarities and distinctions on every single tumor, and that's why it becomes important to do the molecular profiling of the tumors that come out, so that we can actually determine just how close the established models are to the original tumor. This is an example of kind of precision medicine approach we're working on with Andrea's group. doing RNA-guided precision medicine based on single-cell analysis. And so in this case, the first passage, initially implanted tumors were profiled by single-cell RNA sequencing. We used that data to combine with the OncotreAT technology that Andrea has developed to predict different drugs that would target different cellular populations within the tumor. And so this is just a first slide showing how we could identify the epithelial cells specifically, they're in red here, and then focus our analyses just on those red cells. And when we just look at the epithelial cells, we see two major clusters of cells. They align with two of the three major cell types that we have identified across large numbers of human pancreatic cancer single-cell data sets, again, in collaboration with Andrea's group, and then predict cells that match these populations, or predict drugs that match these populations. And so as an example, one of the drugs that we predicted for this group of cells here is a drug called plicamycin. And when we treat it, we then expand the next generation of tumors in the passage 2 and treat an example of those cells, treat those tumors with either DMSO or with plicamycin, you can see that there's a change in the distribution of cells between these two different clusters. And this is after 24 hours of therapy. And so you can argue that either some of the cells in this cluster have died or that they have changed their transcriptional state. I tend to favor the latter interpretation, but we have, this is early days, N of 1 on this type of study. But this gives you an example of the type of work that can be done. And this, again, I think points to the idea that individual drugs so pancreatic cancer is very resistant to chemotherapy, and this idea that that different cellular populations within a tumor may serve as a source of chemoresistance, that the plasticity between these different populations can actually allow tumors to survive insults that would otherwise wipe out the tumor. So that's just one little vignette. I'm now going to switch over to small animal imaging technologies, of which our core facility has 4 that are established as well. And as well, I forgot to mention this, it was back on the first slide, but our core facility also serves as a bridge to the PET center that's run by Akiva Mintz. And they have a mouse micro PET CT instrument in their facility. And they have access, of course, to the linear accelerator there for making various isotopes and tracers. And they've become very good at making kind of experimental tracers. And I would strongly encourage you, if you're interested in this, reach out to our team. We can connect you to Akiva, or you can reach out to him directly. And we can help coordinate, different types of imaging being applied to the same animals, even in some cases. So starting with the first technology, optical imaging. I think this is probably the most familiar form of small animal imaging technology to most biologists, to most cancer biologists. And the idea here is that you are looking at a light source that is either generated locally through a chemical reaction, such as through chemoluminescence, or a light source that is reflected, reflect is not the right word, that results from fluorescence from incoming stimulation of light at a different wavelength. And so the advantage to this technology is that it's pretty high sensitivity, particularly when we're talking about luminescence. But the disadvantage is that it's very low resolution. This is really a functional imaging technology. It's not designed for anatomical imaging, though there are other variants of optical imaging that can be used for that. In this case, the real advantage is the ability to build functional reporters that tell you something about the biology of the tumor or about the biology of the tissue. And so because proteins can be engineered that are luminescent or fluorescent or even both, you have the ability to hook those up and engineer them in very sophisticated ways to ask biological questions about your system. I want to make one note right now, which is that imaging reporters have been undergoing a tremendous revolution recently, making them brighter, more specific, more multifunctional. And as a result, there are a lot of technologies that are well beyond the standard luciferase cassette of the 1990s era. And so I encourage you to reach out to our team. Please consult in advance on your optical reporter designs, because if you're building something from scratch, you don't want to miss the opportunity to use something that's a lot brighter and a lot more capable than the plasmid that's been sitting in your freezer for the past 10 years. So next, I'm going to switch to micro X-ray computed tomography imaging, or micro CT imaging. Our facility has one of the higher sensitivity micro CTs commercially available. We can create anatomical images now at a resolution in the range of 10 micrometer voxels. And just to give you a sense of the kind of resolution that can be achieved, on the top right, this image is the trabecular bone at the head of a long bone in a mouse, I believe femur. And you can see actually kind of this spongiform structure of bone in cross section. And so we can see very, very small structures with this instrument. We also have the ability to use contrast agents, such as the one on the right, or on the left, that allow us to see angiogenesis and other structures. In this case, we were able to distinguish bone in green, the tumor, a pancreatic tumor in yellow, and vasculature in red, and show how this tumor is kind of nestled in within vasculature, but in fact, it's the tumor itself has very few functional blood vessels. There's another image from the micro CT instrument showing both bone in yellow and also in orange, kind of in red-orange color, is vasculature, again, using a blood pool contrast agent, right? With this kind of maximum intensity projection, gives it a 3D view. We also have the ability to use a contrast agent that's taken up by Kupffer cells in the liver, and then you can run a subtraction between the contrasted area of the liver and the non-contrasted area, and actually identify areas that have liver metastases. And this is something that an undergraduate in my team has actually been developing the methodology for, and he's working on writing a small methods paper on this. And this is working pretty well for us right now. So if anyone is interested in liver metastasis imaging, we're able to do this longitudinally over time. I should mention that this CT instrument, because it's so sensitive, is in fact very low dose for a given image. And so even using successive doses, the cumulative dose of radiation that is delivered to the animal is very modest, and as a result should be subtherapeutic, in the subtherapeutic range. Switching now to high resolution ultrasound. And it's funny, within the imaging community, for whatever reason, cross-sectional imaging tends to be more respected than something like ultrasound. And I gotta say, I absolutely love ultrasound. I find it to be one of the most helpful and useful imaging technologies, and also very versatile and facile in its applications. And so it can be used for simple things like anatomical imaging of kind of soft tissues in the abdomen, the heart, or muscle of an animal. We did a pilot study on cancer-associated cachexia, and we were able to show the wasting of muscle in the legs over time in animals as they were developing pancreatic tumors, as an example. And we can mount that two-dimensional transducer onto a three-dimensional motor arm, take multiple sections through the tumor, reconstruct them into a 3D tumor volume, and then that's quite quantitative. And so we can measure this, the growth of a tumor that's even very deep inside the abdomen over time in a fully non-invasive fashion. There are also functional applications for ultrasound. As an example here, this is an anatomical image. I'll set it in motion in just a moment. An anatomical image of a mouse with mouse's abdomen with a pancreatic tumor kind of embedded in the mesentery along the intestines. And what I'm going to do now is switch over to a different mode of imaging called nonlinear contrast ultrasound that really subtracts almost all of the signal. And then what we're going to do is inject a contrast agent called microbubbles, which are gas-filled liposomes that are approximately the size of red blood cells. So they're quite large, and they're not going to escape the vasculature at all. So it's a pure blood pool contrast agent. And as a result, you can visualize tissue perfusion this way. And so as that contrast agent comes into the tissue, you can see really great vasculature in the intestines all around that tumor, but the pancreatic tumor is actually very poorly vascularized, and this is an established feature of the biology of pancreatic ductal adenocarcinoma. This type of image actually was utilized in discovering this concept and really establishing it both in mice and later in humans as well. In fact, to be fair, radiologists have been diagnosing pancreatic cancer as a poorly enhancing tumor, lesion in the pancreas for the past century or so. But this kind of, I think, demonstrates it very nice with in mice. Finally, for ultrasound, we also have the ability to do very high frame rate acquisitions of things like the heart while it's beating. And so this is a slowed down video of the mouse heart beating, and you can see blood flowing through one of the ventricles here. And I'll just set that going again. And you can actually see the muscle contracting and relaxing. And then we can do all sorts of strain measurements and muscle wall thickness measurements and other cardiac functional measurements on this type of image. So there's a lot that can be done to turn these images into quantitative functional information about cardiac health and other types of blood flow within major blood vessels as well, strain imaging, etc. So there's a lot that we can do, and the only way we can help you do it is if we know what the research questions are that you're trying to ask. Finally, this is the 9.4 Tesla MRI on the 10th floor of the Cancer Center for mice. It's designed for mice and maybe the heads of rats. You could maybe possibly squish a fairly youngish whole rat inside the bore, but it's really not designed for that unless we get a different coil in place. But the goal here is mostly for mouse. So this is a 20 centimeter diameter, including all of the equipment that needs to go in there. We can get really pretty anatomical images off of this. So this is a T2 weighted image of the head of a mouse. You can see beautiful anatomic morphology on the cerebellum and other parts of the brain. We can see the cartilaginous rings of the trachea on this mouse. So there's a lot that can be learned from anatomical imaging. I just like this image. I mean, yes, those are the eyes and the brain. But we can also get vast, functional information. This is an angiography study of a mouse with some, malformations. And there's all sorts of different types of functional imaging that can be run, including fMRI, which is technically functional MRI, but really what it's looking at is the blood flow in different vessels within the brain. And we've done a fair bit of this working with our neuroscience colleagues. This is just another couple of images from angiography where you can see some beautiful structural studies of different components of the vascular system. Cardiac imaging, higher resolution even than the ultrasound, and one can do a lot of functional measurements here with the MRI as well. The difference is speed and throughput. With ultrasound, you can get through a lot more animals. So again, it really depends on exactly what it is that you're trying to learn. I will point out that some of these images, like this one over here, has a contrast agent built in, so you can see the blood pool here. Diffusion weighted imaging, which really actually digs down into the diffusion of water and other the diffusion of water in tissues, and actually allows you to measure that spatially and map it out. And there's a lot that can be done from a research standpoint in terms of addressing specific questions. And then finally, the modality that I'm going to end on is magnetic resonance spectroscopy. And the reason I wanted to end on it is because I think that this is probably one of the most critical technologies for our MR center in terms of being useful and providing a very unique and rarefied advantage to cancer center investigators. So I think most folks probably know that the full original name of MRI was actually NMRI, for nuclear magnetic resonance imaging. And they got rid of the nuclear because when it was developed, the idea of nuclear anything was a little bit scary to the general public. But the nuclear is kind of important because it means NMR, right? So this is an imaging version of NMR. And so NMR is used to perform some types of metabolomic studies. And it turns out that you can do metabolomics and look at specific metabolites in animals while they're still alive using high field MRI instruments like the one that we have upstairs. And so that the range of molecules that one can image this way and detect is expanding rapidly over time, and there are some very interesting techniques and some great collaborating investigators here at Columbia who are helping us build out that set of molecules. Among those of interest, we can detect some lipids, we can detect glutathione, and I just wanted to show you one little vignette from my own research. So we recently published this paper studying cysteine metabolism in pancreatic cancer, and this is just a small little excerpt from it. At the end, we showed that there was a drug that was published a couple of years ago called inase that can actually digest cysteine in circulation in mammals. And we showed that in cell culture that it induced a form of non-apoptotic cell death called ferroptosis that I think many of you know was discovered here at Columbia by Brent Stockwell. And what we were able to do was show that this drug then in our genetically engineered model of pancreatic cancer would actually cause tumor stabilizations in these mice over a short period of time. And when we looked in the tumors, we saw histopathological features that we've come to associate with ferroptosis based on work that was earlier in the same paper. And that includes, and as well, it caused, induced tumor necrosis, both microscopically and macroscopically. But we were stuck with this problem of how one proves that a phenomenon that you view, that you visualize by histopathology ex vivo after you've taken a sample out of the mouse is actually in vivo manifestation of ferroptosis, which is a process that's almost entirely been studied in vitro. And so we did our best. We showed that there was accumulation of this lipid oxidation byproduct called 4-hydroxynonenal, that there was not an increase in cleaved caspase 3, that there were the accumulation of very large lipid droplet structures that are associated with lipid oxidation response, that there were mitochondrial defects, but these are all ex vivo analyses. And so one of the things that I think is actually most convincing didn't even make it in the paper, which were down-regulated in mice that were treated with cysteinase. And we were able to do this as a relative measure. So we got a pre-treatment, a pre-treatment image by magnetic resonance spectroscopy, started treating, and then waited, I think it was 7 days or 10 days, I think it was 10 days, and then got a post-treatment image on the same animal. And by having that paired sample structure, you get rid of a lot of the background noise and heterogeneity that's built into these kind of in vivo studies. And that was, that allowed us to kind of really reveal this downregulation, at least in a subset of animals. And it turns out in the animals that had the largest histopathological response post hoc when we looked at their tissues. And so this is, it is early days on spectroscopy, but we have an MRI physicist in the laboratory, Dr. Yanping's son, who has been investing heavily in building out this technology, and particularly from the analysis side, and as well our collaborators such as Christoph Yukim and others who have built some of the, some of the analytical pipelines for looking at these, for looking at spectroscopy data and for analyzing it. So I think this could turn into something that is useful to many, many groups in the Cancer Center. And if you are interested in it, please let us know, because we're starting to build applications into center grants that would include components of magnetic resonance spectroscopy. And we want to make sure that we list as many potential users as possible. And also we'll help start, we'll start helping you look into whether or not it's feasible for your particular metabolite of interest. All right, with that, I am a little bit over time, so I'm going to cut out now, and we'll go ahead and take questions. I'm going to stop my screen sharing now.
Okay, great. Thanks very much, Ken. So please feel free to unmute yourself or put your question in the chat. A question to start off, Ken, about the PDX models and how stable they stay over time compared to the original primary tumor and your experience in that. And also if you've looked at recurrent tumor versus primary tumor in PDX mouse models.
So the answer to the second question is no, it's very early days and we need more users to request the service so we can answer some of these questions. But in terms of the stability of the phenotype, what I can tell you so far is that on the small number of tumors that we've looked at, the regulatory, there's a great deal of enrichment at the regulatory, transcriptional regulatory level as measured by master regulator analysis using Andrea's technology. And so that seems to be stable, at least for the first 2 passages, but we need to take it out further. And again, we're hoping that we get enough users or external support in order to be able to build out a fairly sizable tumor bank that would allow us to really address some of these questions in a more rigorous way on large scale with lots of numbers of tumors. So I'd love to have, a bank with 100 different tumors in it where we could ask exactly those questions.
Great, thank you. And another question, are there any tumors that in your experience don't work with that type of technology or that don't lend themselves to the
Well,
PDX?
so far we've been very fortunate in that all of our attempts to make tumors have been successful, including 2 uveal melanomas, which I was a little bit skeptical of. But that said, we've mostly focused on pancreatic tumors so far, and those are highly engraftable tumors. Historically, the field has found that neuroendocrine tumors in particular have a low take rate, but we're willing to give that a shot, and particularly with this orthotopic approach. I think there may be some relevance, not to try to put those tumors under the skin, which presents a very aberrant local microenvironment. So we're willing to give those a shot, and we do, there is a cost to an unsuccessful attempt to make a xenograft, but it's not nearly as high as a successful attempt. So we kind of absorb some of the cost for unsuccessful ventures, but we're willing to take a shot for almost anything.
Great, thank you. Thanks for those questions. Does anybody have any questions, or unmute yourselves or keep sending the questions to me on the chat? Let's just give people a couple of minutes here. I guess I have a question for you, looking at the last stuff that you put up there. How does that fit in with metabolomics and the type of things that go on in lipid metabolomics? How would those 2 come together?
Yeah, it's brilliantly complementary. So if we have from matched image spectroscopy data set, if we also have metabolomics that have been analyzed by proper NMR, that tremendously increases the power of our ability to interpret our own data set. So let me be very clear. The sensitivity of in vivo magnetic resonance spectroscopy is not nearly what you would get using proper analytical NMR or mass spectrometry or other technologies. The physics of it just doesn't work out, and so it's actually perfect if you have another lipidomic technology, because then you can align the 2 data sets and help identify some of the peaks that are confusing in the MRS data set.
Great, thank you. Anybody have any more questions? Please feel free to unmute yourself while you're thinking or dropping them in the chat. I'm going to share my final slide, and then we can see if anybody has any other questions. If you do have questions in follow-up and you want to contact Ken or Chris or any of the other individuals in OPTIC, please do. I guess, Ken, you guys are ramping up and coming back up to speed at some point over the coming.
We've actually already started in anticipation of the proper phase 2 opening. We've started ordering animals and building up the mouse colonies. I didn't really make a slide about it, but we actually breed our own NRG mice as recipients. And sometimes when we have excess, we'll even provide those at cost to users who want them, which is a lot cheaper than purchasing them from Jack's Labs or whatever.
Great. Okay, thank you. Okay, so we'll wrap up then. Just very quickly let you guys know that we had a seminar scheduled for tomorrow, but we have rescheduled it. We have moved it. Tomorrow there is an initiative that sprang up in the last few days called Shutdown STEM. It is a request, an effort, an initiative to cancel or replace standing seminars or meetings or classes, business as usual, and replace those with discussions with colleagues about anti-Black bias in the world and in academia. The website here for Shutdown STEM is available to you guys, and I can send it on if you like. There are 2 events that we know of tomorrow that may be something you're interested in joining, and I can send anyone the email links for those. I will just put my uni in here into the chat, and please feel free to email me if you'd like more information. I have links to register and join. One is about race and public health, and the other is Vigil through CUMC leadership to Mourn Black Lives and Act to End Structural Racism. So I look forward to joining both of those and to continuing the conversation. Next Tuesday, we will hold our rescheduled Thriving Online Science in the Public Eye, and you'll see more messages about that as the week continues. Meanwhile, thank you very much, Ken. A wonderful seminar, and really enjoyed your presentation today. And thanks to everyone for joining us, and enjoy the rest of your evening.
Bye.
Thanks, guys. Bye-bye.